Hello guys, today we are gonna learn about chromosomes. So the first visible appearance of chromosome or first time the chromosomes were talked about by Strasburger in 1875. So Strasburger in 1875 discovered the thread-like structures which appear during the cell division. So as Strasburger was studying cell division, he just observed the thread-like particles and he named it as a chromosome. So like after a year of finding the or discovering the chromosome, Wilder in 1888 termed the structures as a chromosome. So chromosome is chroma is called color and zones are body. So color imparting bodies are called as chromosomes. So chromosomes shows a basic affinity to the dyes and could be stained deeply, while cytoplasm look little or no stain. And chromosomes are invisible during interphase but are visible during cell division and main the phase where we can literally observe every chromosome is at the time of mitosis and the phase is called as metaphase. Okay, so at the time of metaphase we can literally observe all the chromosomes which are aligned at the center of the cell, and where we can literally watch spindle fibers which attracts or separate the chromosomes body. Okay, then after that the chromosomes are assumed to acquire various shapes and sizes according to the stages of cell division. So at the time of when we observe the normal cell, the chromosomes are thread like structure, but as the cell begins to have the what we can call as the cell begin the cell division, there are different forms of chromosomes we can observe. So first of all we can literally discuss about the two main activities or function of chromosomes. So there are main two functions of chromosome. The first one is to transform the genetic information from generation to generation and from cell to cell. This all happens during cell division. And the second main activity or function of the chromosome is gene expression or the conversion of genetic information for cellular function and development. Okay, so what does gene expression mean? So as we can all see, the gene is nothing but the is a linear form of amino acids, and if we are not able to read it properly, we can't literally express how the gene is to be formed. So there are lots of genes, and there are lots of triplets. So as the Rana has discovered the triplets, every triplet has its own amino acid. So if the amino acid is not being read properly, so there will not be the proper gene expression. So chromosome here are working models which helps in gene expression on, and after that, on the basis of absence and presence of the nucleus living organism. Okay, okay, fine. So this is all about chromosome. So we are gonna see the basic difference between living organisms. So as we can see, the earth has been formed from 4 billion thousand years. So there was big bang theory and all the theories. So the most what we can call it, the most accurate theory which is for the evolution is like what we can call the theory of evolution. Okay, so according to that theory, there are two types of organism, which one are called as prokaryotes and another are called as eukaryotes. So how these cells are being defined, or how these two types of organisms are being differentiated. So on the basis of nucleus, the cells and the humans or the animals are being differentiated into two types. One is eukaryotes and other is prokaryotes. So prokaryotes are the organisms which are having a less developed nucleus, or in some cases the nucleus is absent also. So as we can conclude that there is no nucleus, it doesn't mean that they don't have chromosome. They have chromosomes, but the chromosomes are not linear. The chromosomes in prokaryotes are generally circular or like a single threads. Okay, then there comes eukaryotes. So eukaryotes are the complex organism. Like we can consider all the, like all the sea animals which lives deep down the sea, and all the animals, like we can consider ourselves as eukaryotes. Why we are being called as eukaryotes? Because we are having the specified cell structure. Those our cells undergo cell cycle, and it has specified amount of time which is being taken to convert one cell into another cell. So these are the two main types of cells, that's called as prokaryotes and eukaryotes. Then we will see the basic difference in prokaryotes and eukaryotes. The first and main difference is being presence and absence of a well developed nucleus. So in prokaryotes we can see the nucleus, but it is not that much well developed. But in eukaryotes we can see the well developed nucleus. We are having a nuclear membrane, we are having a nucleolus, we are having lots of other organelles also inside the nucleus. So this is one of the main difference. Then in prokaryotes the genome is not included in the nuclear envelope. Yeah, as I have earlier told you, we, like as a eukaryotes, are having nuclear envelope, but in prokaryotes there is not no any nuclear envelope. And the main another difference is genetic material is present in nuclear which contain the highly folded circular DNA. As we have earlier mentioned, the DNA which is found in prokaryotes is circular, while in humans it is a linear form, and it is highly, like what we can call, it is highly condensed. Then in prokaryotes we can see like bacteria, blue green algae, red algae, and in eukaryotes we can see all the animals other than that algae. So after that, in prokaryotics, the cell division is amitotic. It means that they are having binary fissions or another kind of cellular, what we can call it, like cellular divisions. But in humans we can see the complex processes like mitosis and meiosis. So this is one of the main difference in prokaryotes and eukaryotes. Then in prokaryotes generally the cells or generally the organisms are in haploid stage, but in eukaryotes our cells are diploid, as we can only consider the uh, female and male sex cells are only the haploid cells in all the humans. But in prokaryotes all the cells are haploid. So this is one of the main difference. Then genes and proteins products are collinear. It means that there is point by point relationship between the codons and DNA and corresponding amino acids in the peptide chain synthesized. Split genes are extremely rare in prokaryotes, but in eukaryotes many genes are splits or interrupted, as we can see the phenomena like multiple genes and like a linkage. All the phenomena are seen only in eukaryotes and are absent in prokaryotes. So these are some of the main and major differences of prokaryotic and eukaryotic cells. So as we can see, the main difference is according to the chromosome. So we will see the structure of chromosome, that is eukaryotic chromosome. So as we can see, if you ask me if I'm being able to see the chromosome in an interphase or in another phase of of cell division, so the answer is clearly no. I can see the cells only at the time of literally cell division. So what does cell division mean? So cell division composed of three parts. It's S1, G phase and S2 phase. So S1 is synthesis phase, and G is the phase where actual mitosis or meiosis takes place, and S3 is the phase where the cell are reformed and are being what we can and are being splitted toward the different organisms. So chromosomes are more clearly visible during mitotic metaphase, as I have earlier mentioned it. So metaphase is the best stage of cell cycle where we can study or we can watch or see the chromosomes. Then the chromosomes bear the genes in a linear fashion and are concerned with transmission of characters from generation to generation. So if as we all know that as a human we are kind of similar to our parents. The 50% of our mother's genes and 50% are of our father genes. So how they are being transmitted. So all of these transformation processes are take with the help of chromosome. So what we call DNA is a chromosome. So with the help of chromosome we can like transfer. Hereditary and variations are the main functions of chromosome. Okay, then chromosomes of eukaryotes are enclosed in a nuclear membrane. They are well protected, and they are well what we can call, they are they are well protected in the humans. As you can see, there are lots of problems which happen when the chromosomes are not in a proper in a proper number, or in a proper like what we can call like a proper structure. If in case any structure is being deteriorated, or any number of chromosome is lesser, or being increased, the humans are more are more being suffered by the different types of diseases. So there are lots of diseases which are caused due to deletion, translocation, transmission of the chromosomes from one place to another. So we will see it later. Then after that we can see, and we can came to a conclusion that chromosomes vary in shape, size and number in a different species of plant and animals. Just can we can see wheat is a hexaploid, and humans are diploid, and there are lots of species of banana which are triploid, and the watermelon which are triploids are seedless. So these are the types of or number of chromosomes which vary in species to species. And as the number of chromosomes vary, the characters which are imparted, or the characters which are being shown by the individuals also varies with the number of chromosome, as well as structure and size of chromosome. So chromosomes are of course composed of DNA, RNA and histone. Histone are the proteins which bind DNA, RNA together to make a complex structure which we called as chromosome. After that we will see the morphology. So what does morphology means? So as we all can see, every individual or everything which can be seen or which is visible have specified kind of characteristics. So there are different, or there are characteristics of chromosomes which are to be studied. So the most important character of a chromosome which is eukaryotic is that it is covered, or it is included, or it is bounded by nuclear envelope. So wherever you observe a chromosome which is included, or encloses by the nuclear membrane, it is to be concluded that it is the eukaryotic chromosome. Okay, after that there are two pairs of homologous chromosome in a diploid species. So as we can see, the chromosome is double stranded structure. So one strand is homologous to another, and both the strands are called as sister chromosomes. Okay, after that but we can if I have to study all the chromosome, where can I being study that thing? So as we have all know that we can study chromosomes at the mitotic metaphase. Okay, so mitotic metaphase is one of the most important phase in a cell cycle, where I can or you can observe the chromosomes. So what exactly chromosome is and how we can observe it? So whenever we literally learn anything, so we have to cut it down in two parts. So chromosome composed of two words. It's one is chromo or chroma, and another is zome. So what is meant by chroma? Chroma, it means color, and SOM is zome, means someone which can impart the color. So chromosomes are the organelles which can impart the colors. So if I have to observe the chromosomes, I would use dyes. Okay, so there are lots of dyes which will make my chromosome stay, and I will be able to watch them under the microscope. So what are the different types of what are the different types of structures or features which can be seen under the microscope of chromosome? The main and most important thing which a chromosome consists is chromatid. So chromatid are the two linear strands which are being holded together with chromosome or chromomere. Chromomere is a circular part which hold two chromatids. Okay, then there are secondary constrictions or satellite. Then then comes knobs. Then there are telomeres, then chromomeres, and then chromonema and matrix and pellicle. So what are the chromatids? Chromatids are the two strands which are being included or which are being put together by centromere. Okay, so centromere is a complex part where where the chromosome is highly like where the chromosome can be highly strained. Okay, after that the function of, we can see the function of chromomere. The chromomere are responsible for the shape of chromosome. So by the position of chromomere or centromere, there are different types of chromosomes. The first one is metacentric. So in metacentric, the chromo or centromere is at the center. Then at submetacentric, it is slightly above the center. Then telocentric, it means that it's near to the end, and acrocentric, it means that it is like at the end. So these are the different types of chromosome which are being differentiated on the basis of centromere. After that we can see the secondary constriction or satellite. The secondary constrictions are same that of centromere, but they are not really centromere. So these are the regions where like the what we can call the centromeres are being, or the chromatids are being differentiated or differentiated in another position. After that we can see the knobs. The knobs are the spherical heterochromatic bodies which are present on a chromosome. After that telomeres. Telomeres are the end, like the end positions of chromatids are called as telomere. Then chromomeres. Chromomeres are the are seen at during the early prophase stage, and they are observable with compact structures, and this can be, we can see them after staining practices. After that we are having chromonema. Chromonema are the, under the light microscope, a number of longitudinal strands that can be observed in a chromosome. These strands are called as chromonema. Singular strand is called as chromonemata, and in plural we call it as a chromonema. So chromonema is composed of chromatin and contains gene, and it is not uniform in thickness. So what exactly the most important part in a chromosome? It is chromonema, as chromonema consists of DNA and the information which we have to transmit from one generation to another. Then there are matrix and pellicles. So matrix and pellicles are just like the protecting structures which are important in a structure of chromosome. If that much of genetic material is not being properly protected, it can cause lots of deformities in humans. So these all structures are to be like protected. So there comes matrix and pellicle. Then there are different special types of chromosome. So the main chromosomes are nucleolar chromosomes. The first and main important type of chromosome is a nuclear chromosome, which all of us possess, which all which are having most and foremost importance in a human or in a history of living beings. Okay, after that there are nucleolar chromosomes. These are the chromosomes which are present outside of the nucleus but in a nucleolar membrane. So they are not that much of important, but the nucleolar chromosomes which are having the ability to transform from one generation to another are mitochondrial chromosomes. So these are the most important type of chromosome which play important part in the breeding programs also, as we can see CGMS, GMS. So here are the chromosomes which are more important in that kind of processes are the nucleolar chromosomes. After that we are having sex chromosomes. So these are the most important, as we can see the like as a humans we are having two sex, male and female. So what is the main difference? So as we can see, females of humans are having XX chromosome and males are having XY chromosome. So it is the difference of only one chromosome which literally decided all the different characteristic and all the different things which we possess as a woman and man. So this is one of the most important type of chromosomes. It is called as sex chromosome. So there comes a lots of stages or lots of like deformities where like these chromosomes didn't work, and so we can have like literally another kind of genders which we like openly concluded as transgenders. After that, we are having holocentric chromosomes. So, in general, centromere is located on the plant and animal chromosomes, and one centromere per chromosome is one of the most common situation which is observed. But in certain animals and plant species, the centromeric activity is spread all over, and it is like the whole like chromatid, जो the chromatid whole chromatid like line is like considered as active. So these like centromeres or these chromosomes are called as holocentric. It means that they will be like we can see all the nuclear activities all over the chromosomes. So these are one of the main types of chromosomes. After that, we can see B chromosomes. So B chromosomes are the particular kind of supernumerary chromosomes that may be missing in organisms or may be found as extra chromosomes over and above the standard diploid or polyploid chromosomes, complement consisting of autosomes or a chromosome including sex chromosome if any. Okay, so the B chromosomes are found in the natural populations of many species, both plants and animals. So B chromosomes were firstly reported in maize by Randolph, and B chromosomes have been reported in more than 1000 species of plants. So these are also one of the most important types of chromosome. After that, B chromosomes are recognized on the basis of the following characteristic. They are dispensable and are not be found in all the individuals. So we can like if I am having B chromosomes, if you observe it sometimes at the place of my heart, and you will not be able to find it another time at my heart, or it can be like disposable or dispensable. It like it changes its position. It's just like a transgene. Okay, so after that. They are not homologous with any of the basic A chromosome, and their inheritance is non-Mendelian. It means that they cannot like you can't guaranteed the presence of B chromosome in a like siblings or in a what we can call like in a offsprings if they are even present in parents. So this is one of the most important thing which can be like concluded from B chromosome. Okay, then there are two types of B chromosome. There one are stable and another are unstable. So the stable B chromosomes are mitotically stable, and all the cells of an individual have the same number of B chromosomes. And in case of unstable, the B chromosomes of this group are mitotically unstable and give rise to the cells with different number of B chromosomes within the same individual. Okay, after that, B chromosomes are of different types that are standard type, small types, very small type, and large type. After that, we can see the polyteny or giant or salivary gland chromosomes. So these are the special kind of chromosomes which are observed in a salivary glands of the dipteran larva. Okay, so the polyteny giant chromosomes are typically found in salivary gland cells of the dipteran larva, and they are been found in intestinal cells of mosquitoes. Okay, so this is one of the important important organism we have to keep in our mind, that the polyteny cells or the like salivary gland cells are intestinal cells which are found in mosquitoes. Okay, after that, in plants polyteny chromosomes have been reported in nutritive tissues such as suspensor cells of a kidney bean. Okay, so here are two important like examples. One is mosquito and another is kidney bean. So the polyteny or giant chromosomes are found in mosquitoes as well as in kidney beans. Okay, after that, the that these type of chromosomes are observed and are discovered by Balbiani in 1881. So there comes a like whenever we like study the structure of this, so there are the types of rings which are present all over the B chromosome. So they are called as Balbiani rings. So why they are called Balbiani rings? Because they are being discovered by the scientist called Balbiani. So these are the different types of chromosomes, and this is like all about the chromosome we can conclude. After that, we will see. After that, there is also one last type of chromosome which is called as lamp brush chromosome. So lamp brush chromosomes were first discovered in shark in 1892 by Puckett. Okay, so like as we can all see, the humans are very like interesting or like very enthusiastic creatures, so they study all the individuals. So as the human being studied shark, they came to know that there comes a different types of chromosome which are not present all over the individuals, or they are not being found in anywhere in another organism. So these are the cells which are found in sharks. So these cells are called as lamp brush cells. So they are having a structure like lamp brush. So they are having a like they are having two types of structure. So they have chiasma, they have loops, they have centromere, and their visible structure consists of loop matrix, loop axis, and central axis, as well as chromomere. So chiasma is always located between loops but never within a loop. Each loop contains a single DNA of a double helix. Okay, so these are the different types of like chromosomes which can be studied and which can be seen. So after this, we will see the deformities or the like diseases which are caused due to like breakage of chromosomes, or due to like less or more numbers of chromosomes. So yeah, thank you.